Diatoms are a major group of algae, and are among the most common types of phytoplankton. Diatoms are unicellular, although they can form colonies in the shape of filaments or ribbons, fans, zigzags, or stars. The first diatom formally described in scientific literature, the colonial Basilaria paradoxa, was found in 1783 by Danish naturalist Otto Friedrich Müller. Diatoms are producers within the food chain. A unique feature of diatom cells is that they are enclosed within a cell wall made of silica called the frustule. These frustules show a wide diversity in form, but are usually almost bilaterally symmetrical, hence the group name. The symmetry is not perfect since one of the valves is slightly larger than the other, allowing one valve to fit inside the edge of the other. Fossil evidence suggests that they originated during, or before, the early Jurassic period. Only male gametes of centric diatoms are capable of movement by means of flagella. Diatom communities are a popular tool for monitoring environmental conditions, past and present, and are commonly used in studies of water quality. General biology. There are more than 200 genera of living diatoms, and it is estimated that there are approximately 100,000 extant species. Diatoms are a widespread group and can be found in the oceans, in fresh water, in soils and on damp surfaces. They are especially important in oceans, where they are estimated to contribute up to 45% of the total oceanic primary production. Spatial distribution of marine phytoplankton species is restricted both horizontally and vertically. Though usually microscopic, some species of diatoms can reach up to 2 mm in length. Diatoms belong to a large group called the heterotrophs, including both autotrophs and heterotrophs. Their yellowish-brown chloroplasts are typical of heterotrophs, having four membranes and containing pigments such as the carotenoid phlocoxanthin. Individuals usually lack flagella, but they are present in male gametes of the centric diatoms and have the usual heterotroph structure, except they lack the hairs characteristic in other groups. Most diatoms are non-motile, as their relatively dense cell walls cause them to readily sink. Planktonic forms in open water usually rely on turbulent mixing of the upper layers by the wind to keep them suspended in sunlit surface waters. Some species actively regulate their buoyancy with intracellular lipids to counter sinking. A feature of diatoms is the urea cycle, which links them evolutionarily to animals. This was discovered in research carried out by Andrew Allen, Chris Bowler and colleagues. Their findings, published in 2011, that diatoms have a functioning urea cycle was highly significant, since prior to this the urea cycle was thought to have originated with the metazoans who appeared several hundreds of millions of years after the diatoms. Their study showed that while diatoms and animals use the urea cycle for different ends, they are seen to be evolutionarily linked in such a way that animals and plants are not. Diatom cells are contained within a unique, silica cell wall comprising two separate valves. The biogenic silica that the cell wall is composed of is synthesized intracellularly by the polymerization of silicic acid monomers. This material is then extruded to the cell exterior and added to the wall. Diatom cell walls are also called frustules, or tests, and the two valves typically overlap one over the other like the two halves of a petri dish. In most species, when a diatom divides to produce two daughter cells, each cell keeps one of the two halves and grows a smaller half within it. As a result, after each division cycle, the average size of diatom cells in the population gets smaller. Once such cells reach a certain minimum size, rather than simply divide, they reverse this decline by forming an auxosphory. This expands in size to give rise to a much larger cell, which then returns to size-diminishing divisions. Auxosphory production is almost always linked to meiosis and sexual reproduction. Decomposition and decay of diatoms leads to organic and inorganic sediment, the inorganic component of which can lead to a method of analyzing past marine environments by corings of ocean floors or bay muds. since the inorganic matter is embedded in deposition of clays and silts and forms a permanent geological record of such marine strata. The study of diatoms is a branch of phycology, and phycologists specializing in diatoms are called diatomists. Classification. The classification of heterozoa is still unsettled, and they may be treated as a division, kingdom, or something in between. Accordingly, groups like the diatoms may be ranked anywhere from class to division, with corresponding changes in the ranks of their subgroups. Diatoms are traditionally divided into two orders: centric diatoms, which are radially symmetrical; pinnate diatoms, which are bilaterally symmetrical. The former are paraphyletic to the latter. Due to the difference noted in pinnate diatoms, of the presence or absence of a rapha, a more recent classification by Round, Crawford and Mann divides the diatoms into three classes and a number of orders. Class Cosynodiscophyceae. Round and R.M. Crawford. Centric diatoms Anorales. Round and R.M. Crawford. Arachnoidiscales. Round. Astralamprales. Round. Aulicocerales. R.M. Crawford. Bidophilus Chaetocerotula. Round and R.M. Crawford. Chrysanthemodiscales. Round. Corythrales. Round and R.M. Crawford. Cosynodiscales. Round. Cymatocerales. Round and R.M. Crawford. Ethmodiscales. Round. Hemiorlales. Round and R.M. Crawford. Leptocylindrales. Round and R.M. Crawford. Lithodesmialis meloceralis. R.M. Crawford. Orthocerales. R.M. Crawford. Paraleales. R.M. Crawford. Rhizoseleniales. Stictocyclales. Round. Stictodiscales. Round and R.M. Crawford. Thalassiocyrales. Triceratialis. Round and R.M. Crawford. It is probable there will be further revisions as understanding of their relationships increases. Medlin and Kaczmarska propose the following classification for the diatoms: Bacillariophyta Cosynodiscophytina Cosynodiscophyceae Bacillariophytina Mediophyceae Bacillariophyceae. Diatoms generally range in size from 2 to 200 micrometers, and build intricate howard but porous cell walls composed primarily of silica. This siliceous wall can be highly patterned with a variety of pores, ribs, minute spines, marginal ridges and elevations, all of which can be used to delineate genera and species. The cell itself consists of two halves, each containing an essentially flat plate, or valve and marginal connecting, or girdle band. One half, the hypotheca, is slightly smaller than the other half, the epitheca. Diatom morphology varies. Although the shape of the cell is typically circular, some cells may be triangular, square, or elliptical. Cells are solitary or united into colonies of various kinds, which may be linked by siliceous structures, mucilage pads, stalks or tubes, amorphous masses of mucilage, or by threads of chitin, which are secreted through strutted processes of the cell. Major pigments of diatoms are chlorophylls A and C, beta-carotene, fucoxanthin, diatoxanthin, and diadinoxanthin. Diatoms are mainly photosynthetic. A few, however, are obligate heterotrophs, while others can live heterotrophically in the absence of light, provided an appropriate organic carbon source is available. Storage products are chryslaminarine and lipids. HOK-Al also provide a comprehensive coverage of diatom taxonomy. Ecology. Planktonic diatoms in freshwater and marine environments typically exhibit a boom and bust lifestyle. When conditions in the upper mixed layer are favorable, their competitive edge allows them to quickly dominate phytoplankton communities. As such, they are often classed as opportunistic strategists. The freshwater diatom Didymusphenia geminata, commonly known as Didyma, causes severe environmental degradation in watercourses where it blooms, producing large quantities of a brown jelly-like material called brown snot, or rock snot. This species is native to Europe and is an invasive alien both in the Antipodes and in parts of North America. The problem is most frequently recorded from Australia and New Zealand. When conditions turn unfavorable, usually upon depletion of nutrients, diatom cells typically increase in sinking rate and exit the upper mixed layer. This sinking is induced by either a loss of buoyancy control, the synthesis of mucilage that sticks diatoms cells together, or the production of heavy resting spores. Sinking out of the upper mixed layer removes diatoms from conditions unfavorable to growth, including grazer populations and higher temperatures. Cells reaching deeper water or the shallow seafloor can then rest until conditions become more favorable again. In the open ocean, many sinking cells are lost to the deep, but refuge populations can persist near the thermocline. Ultimately, diatom cells in these resting populations re-enter the upper mixed layer when vertical mixing entrains them. In most circumstances, this mixing also replenishes nutrients in the upper mixed layer, setting the scene for the next round of diatom blooms. In the open ocean, this cycle of bloom, bust, then return to pre-bloom conditions typically occurs over an annual cycle, with diatoms only being prevalent during the spring and early summer. In some locations, however, an autumn bloom may occur, caused by the breakdown of summer stratification and the entrainment of nutrients while light levels are still sufficient for growth. Since vertical mixing is increasing, and light levels are falling as winter approaches, these blooms are smaller and shorter-lived than their spring equivalents. In the open ocean, the condition that typically causes diatom blooms to end is a lack of silicon. Unlike other nutrients, this is a major requirement solely of diatoms, so it is not regenerated in the plankton ecosystem as efficiently as, for instance, nitrogen or phosphorus nutrients. This can be seen in maps of surface nutrient concentrations. As nutrients decline along gradients, silicon is usually the first to be exhausted. Because of this bloom and bust cycle, diatoms are believed to play a disproportionately important role in the export of carbon from oceanic surface waters. Significantly, they also play a key role in the regulation of the biogeochemical cycle of silicon in the modern ocean. The use of silicon by diatoms is believed by many researchers to be the key to their ecological success. In a now classic study, Egon-Axness found that diatom dominance of mesochasm communities was directly related to the availability of silicic acid, when concentrations were greater than 2 micromol m-3. They found that diatoms typically represented more than 70% of the phytoplankton community. Raven noted that, relative to organic cell walls, silica frustules require less energy to synthesize, potentially a significant saving on the overall cell energy budget. Other researchers have suggested that the biogenic silica in diatom cell walls acts as an effective pH buffering agent, facilitating the conversion of bicarbonate to dissolved CO2. Notwithstanding the possible advantages conferred by silicon, diatoms typically have higher growth rates than other algae of a corresponding size. Diatoms occur in virtually every environment that contains water. This includes not only oceans, seas, lakes and streams, but also soil. Life cycle. This is used by each daughter cell as the larger frustule into which a second, small frustule is constructed. This form of division results in a size reduction of the daughter cell that received the smaller frustule from the parent, and therefore the average cell size of a diatom population decreases until the cells are about 1/3 their maximum size. It has been observed, however, that certain taxa have the ability to divide without causing a reduction in cell size. Nonetheless, in order to restore the cell size of a diatom population for those that do endure size reduction, sexual reproduction and auxosphory formation must occur. Vegetative cells of diatoms are diploid, and so meiosis can take place, producing male and female gametes which then fuse to form the zygote. The zygote sheds its silica theca and grows into a large sphere covered by an organic membrane, the auxosphory. A new diatom cell of maximum size, the initial cell, forms within the auxosphory, thus beginning a new generation. Resting spores may also be formed as a response to unfavorable environmental conditions, with germination occurring when conditions improve. Diatoms are mostly non-motile, however, sperm found in some species can be flagellated, though motility is usually limited to a gliding motion. In centric diatoms, the small male gametes have one flagellum while the female gametes are large and non-motile. Conversely, in pinnate diatoms both gametes lack flagella. Certain aphid species, that is pinnate diatoms without a rapha, have been documented as anisogamous and are, therefore, considered to represent a transitional stage between centric and raphid pinnate diatoms, diatoms with a rapha. Evolutionary history. Heterochloroplasts appear to be derived from those of red algae, rather than directly from prokaryotes as occurred in plants. This suggests they had a more recent origin than many other algae. However, fossil evidence is scant, and it is really only with the evolution of the diatoms themselves that the heterochore make a serious impression on the fossil record. The earliest known fossil diatoms date from the early Jurassic, although the molecular clock and sedimentary evidence suggests an earlier origin. It has been suggested that their origin may be related to the end Permian mass extinction, after which many marine niches were opened. The gap between this event and the time that fossil diatoms first appear may indicate a period when diatoms were unsilicified and their evolution was cryptic. Since the advent of silicification, diatoms have made a significant impression on the fossil record, with major deposits of fossil diatoms found as far back as the early Cretaceous, and some rocks being composed almost entirely of them. Although the diatoms may have existed since the Triassic, the timing of their ascendancy and takeover of the silicon cycle is more recent. Prior to the Phanerozoic, it is believed that microbial or inorganic processes weakly regulated the ocean's silicon cycle. Subsequently, the cycle appears dominated by the radiolarians and siliceous sponges, the former as zooplankton, the latter as sedimentary filter feeders primarily on the continental shelves. Within the last 100 mi, it is thought that the silicon cycle has come under even tighter control, and that this derives from the ecological ascendancy of the diatoms. However, the precise timing of the takeover is unclear, and different authors have conflicting interpretations of the fossil record. Some evidence, such as the displacement of siliceous sponges from the shelves, suggests that this takeover began in the Cretaceous, while evidence from radiolarians suggests takeover did not begin until the Cenozoic. The expansion of grassland biomes and the evolutionary radiation of grasses during the Miocene is believed to have increased the flux of soluble silicon to the oceans, and it has been argued that this has promoted the diatoms during the Cenozoic era. Recent work suggests that diatom success is decoupled from the evolution of grasses. Although both diatom and grassland diversity increased strongly since the Middle Miocene, diatom diversity over the Cenozoic has been very sensitive to global temperature, particularly to the equator pole temperature gradient. Warmer oceans, particularly warmer polar regions, have in the past been shown to have had substantially lower diatom diversity. Future warm oceans with enhanced polar warming, projected in global warming scenarios, could thus in theory result in a significant loss of diatom diversity, although from current knowledge it is impossible to say if this would occur rapidly or only over many tens of thousands of years.